Hello, everyone. Good morning, good afternoon, good evening. Wherever you're joining us from, I'm Ara Khachaturian on behalf of the Campaign to Prevent Alzheimer's Disease, the Brainwatch Coalition, and the International Neurodegenerative Disorders Research Center. Welcome to this inaugural episode of our new series, Enabling Next-Gen Clinical Trial Design with Proteomics and Transcriptomics. Today's episode is titled From A4 to Ahead, Can Proteomics and Transcriptomics Help Us Rethink AD/ADRD Trials? With two disease-modifying therapies now commercially available, Leqembi and Kisunla, we're at a real inflection point. If we want to scale the benefit of early intervention, we also need to invest in early detection, especially for individuals with mild cognitive impairment, subjective cognitive impairment, or even asymptomatic pathology.
Hey everyone, good evening, and I'm Ornit Chiba-Falek from Duke University with the Department of Neurology and Pathology and the Chief of the Division of Translational Brain Science. And yes, Ara, exactly. And if ADRD clinical trials are ever going to reach the population level, they will need detection strategies that work in real-world setting across primary care, underserved communities, and beyond academic research centers.
Which brings us to today's guest, Dr. Paul Aisen, founding director of the Alzheimer's Therapeutic Research Institute at the University of Southern California and a pioneering figure in Alzheimer's disease research for over 3 decades. Paul has led many of the field's most influential trials and has helped shape the methodologies we rely on today. We are honored to have him join us to explore how proteomics and transcriptomics used in tandem with frameworks like ATN and polygenic risk scores might help sharpen clinical trial design and accelerate the shift toward personalized medicine.
So let's start with our corner one. Paul, you have led several landmark studies, A4, Learn Ahead. What insight have you gained that you think the next generation of trialists should really pay attention to?
Well, that's a broad question. Let me see if I can address trial design and lessons from these trials. So A4, Learn, and Ahead all aimed for earlier intervention. And I certainly believe that early intervention is a way to have the greatest impact on slowing progression and the greatest impact on the overall disease. And so the drugs that we have, the 2 drugs that you mentioned, these are approved for the treatment of symptomatic disease. They demonstrate that removing amyloid, dramatic removal of amyloid from brain, can slow disease progression. I think this is clearly proven. But the extent of slowing of disease progression with these drugs is around 30%. I think that's a tremendous benefit that can delay the loss of function or the loss of independence by months or years, depending upon how long the drugs are administered. And that's a huge benefit. But 30% slowing is also obviously not enough. We want to control this disease. And I think that while we can continue to work on trial designs in symptomatic disease, and I think combination therapies are a natural way forward, combining agents that target amyloid with other ideas, drugs that target tau, but also drugs that target other mechanisms, vascular, inflammatory, endocrine, other mechanisms that contribute to the disease as it progresses. I think combination therapy is a natural way to think about increasing the benefit that we've established with these 2 drugs. But another major approach, and the one that's represented by A4 and Learn and Ahead, is treating earlier. So those studies are conducted in a population without symptoms. For the A4 study, we screened people on the basis of age, not on the basis of symptoms. Anybody who was 65 years old or older was eligible, and we screened people with amyloid PET scans And as we expected from experience in ADNI and other studies, 30% of the people we screened had elevated amyloid in brain by PET scan. These individuals were interested in participating in therapeutic trials, even though they did not have symptoms, based on the idea that early intervention in the disease process was the most promising approach to having a dramatic effect on disease progression. Now, the A4 study was a negative trial. The intervention, solanezumab, which is an antibody that binds to monomeric A beta, did not significantly reduce amyloid load, and we knew that it wouldn't. We didn't expect it to. We hoped that targeting the monomeric A beta alone would be enough at this stage of disease. And the answer to that question is no, it was not enough. And solanezumab did not benefit people selected on the basis of amyloid in brain, but no, at the stage before symptoms. That trial was negative. But it taught us a lot. The data from the A4 trial is a huge, rich data set, which combines cognitive and clinical and demographic data with imaging from MR, fMRI, amyloid PET, and tau PET, biofluid assessments with assays of proteins and genetics, just a huge data set that we've now shared with investigators around the world. It's up publicly for sharing, the entire data set. And we've learned a lot. And we've learned, for one thing, that the ideas behind the design of A4 were reasonable. The progression occurred as we expected. The measures that we incorporated into the A4 design were sensitive to decline at this pre-symptomatic stage. Both measures of cognition and measures of subjective concerns were useful. And we have now moved to the AHEAD program, which moves from solanezumab targeting monomeric A beta to lecanemab, which targets protofibrils and fibrils and dramatically removes amyloid, with the idea that using the tools developed and established by A4 and using lecanemab, which we know slows progression by about 30%, if we move into an asymptomatic population, we can increase the disease slowing effect. We know from looking at the trials of lecanemab and donanemab in symptomatic disease that the people who were earlier in the disease process had a greater degree of benefit, a greater slowing of progression. And if we extrapolate into a pre-symptomatic population, we're very optimistic that AHEAD will reveal a greater disease slowing effect than the trials conducted in symptomatic AD. So we're encouraged and we're optimistic. We'll be hearing about the donanemab preclinical study, Trailblazer ALZ-3, pretty soon, and we still have a few years left in the AHEAD trial since it's a 4-year treatment period. But we're optimistic that we're on the path to more effective disease-slowing therapy. That said, the success that we've had to date in symptomatic disease with lecanemab and donanemab, and now the trials in preclinical AD, they're all based on a molecular understanding of the disease. That we've gotten to this stage where we can slow the disease down by going after the specific molecular changes. And those, of course, include aggregation of A beta into oligomers that are highly toxic and protofibrils and plaques, hyperphosphorylation of tau leading to tangles, and many other changes. TDP-43, alpha-synuclein, vascular changes. And we need to bring our best therapeutic approaches to the mechanisms that drive the disease forward, particularly as the disease progresses. It's not just driven by amyloid, it's driven by other proteinopathies and other changes in brain physiology. We have to understand those changes to offer our best therapeutic strategies to control this terrible disease. And so we now have gone from PET scans to blood tests, which is terrific. And we have great blood tests to track amyloid and tau pathology. We still don't have a blood test to track alpha-synuclein or TDP-43 or any of the other co-pathologies. I think that getting blood tests to allow us to identify the presence of other pathologies and to enable us to develop additional targeted therapies for combination approaches, I really think this is a path to a better future for AD therapeutics.
Oh, well, thank you for that answer. You know, I mean, in terms of talking about really getting into the molecular aspects of the disease, I mean, my thoughts were going to the heterogeneity. As you well know, that's a huge problem from a clinical trial design perspective, as well as even thinking about, you know, we're talking about personalized medicine, the need for biofluids, different types of assays, health outcome measures to better help assess who might be at what stage of disease or who might be able to respond well to a particular therapeutic that's coming down the pipeline. What do you see in terms of clinical trial design, for example, with AHEAD, that can really sort of help develop that real world evidence for even those tools that will eventually help primary care and really affect the real world deployment of these interventions?
Yeah, so there are some general principles here that I think we need to keep first and foremost in our minds. These studies are hard and they take a long time and they're expensive. It's a huge effort to do big definitive trials in Alzheimer's disease. And we need to take advantage of everything that we learn. And the way to do that is by sharing and collaborating. So I guess I would start by saying we need to work together. That's how we got to where we are. And that's how we're going to move forward. I hope that we are actually leading the way in terms of data sharing. So as I mentioned, we have shared the entire data set, everything, images, genetics, cognitive and clinical data, everything from A4, ARM codes, everything with the world's research community. And anybody else who wants to get that, it's available for anybody at no cost. We have a very simple process for requesting, and then we rapidly share all the data from A4, and that includes biofluids. And we have specimens and we share specimens, and that allows further exploration of molecular mechanisms and linking them to longitudinal change in the disease. So with this data set, we know we can tell you what's going to happen to every structure in the brain over a period of 4 to 8 years of follow-up. And anything you can get out of any biofluid sample can be assessed in terms of this deep phenotypic long-term study. And that's going to reveal new clues that'll give us new strategies. The AHEAD study is even bigger. And the AHEAD study involved screening 20,000 people. We enrolled 1,600 people. These are all asymptomatic. Part of the reason that we had to screen so many is that we lowered the age range. It's not just starting at age 65, it starts at age 55 when the great majority of people are amyloid negative. But in order to manage such a huge screening process, we developed during the course of the trial an approach that used plasma testing as pre-screening. We developed an algorithm to predict amyloid PET. The algorithm is highly accurate. We screened everybody with blood testing for A beta ratio and p-tau217, an ApoE genotype that allowed us to predict positivity. And we collected information on a total screening sample of 20,000, but we have amyloid PET and tau PET and MRI on thousands of people. And we are releasing all of that data. I know this sounds like a self-advertisement, but I'm talking about how to work together to collaborate to move forward. We're sharing all that data 1 year after the close of the recruitment process. We finished recruitment in October of 2024, and we have pledged and are committed to sharing all of the pre-randomization data. So that's the screening data on those 20,000 people. We're going to release that by October 15th of this year. And I think there will be a lot of clues there and a lot of delving into plasma for indications of other pathologies that ultimately can be linked to the 4 years of study in AHEAD. And once the trial is done in 2028, we will share all that data as well. So I think that we will learn. It's a huge opportunity to understand the disease better, to understand the early stages of disease in particular, to understand which molecular signals in screening plasma predict changes in brain structure, in brain function, in clinical measures over a period of years. And that's going to be hugely helpful in taking us to the next step.
Thank you, Paul. And just to extend on your comment of a molecular mechanism and the linkage to basic science understanding of the underpinning disease mechanism for the AD and ADRD. So technological advancement at the forefront move basic science research forward to better understand disease mechanism and to deepen our knowledge of disease heterogeneity and risk. This understanding, as well as the technological innovation, including a different omics platform, single-cell transcriptomic and single-cell epigenomic, proteomics, and all of this multi-omics platform innovation could and even should be implemented in clinical research and eventually improve patient care. So let's just pivot for lesson learned to where we might go next. And I'm very intrigued to hear your vision of how can we utilize technological innovation and our foreseen deeper understanding of disease heterogeneity and disease risk into a clinical study and drug development for AD and ADRD.
Yeah, so I fully agree that it is a deep study of the basic science behind this disease that will lead to continuing advances in therapeutics. Again, we got here by understanding the role of amyloid. We have a lot to learn about the co-pathologies, the co-mechanisms that drive the disease forward. And I think that we need to all try different methods of studying specimens, scans, and data sets to delve more deeply into the disease process. Are there going to be subgroups that should be treated differently? Yeah, I think there are. I think that we already know that subgroups respond differently, particularly in terms of risk with current therapies. So the number of copies of your ApoE4 allele determine the risk of ARIA events. And already subgroups are being managed differently in some settings. When we're talking about something like a co-pathology, let's consider alpha-synuclein, for example. There is Lewy body pathology in 40 or 50% of people with Alzheimer's disease. that pathology is clearly contributing to disease. People with that pathology tend to progress faster. I believe we need effective therapies against the alpha-synuclein proteinopathy, just as we have effective therapies against amyloid pathology. The alpha-synuclein pathology targeting therapies are only going to be useful and should only be tested and utilized in the subset, close to half of people with Alzheimer's disease who have alpha-synuclein pathology. And so we need to understand what signals in plasma and what imaging modalities will reveal the presence of synucleinopathy to guide our identification of people and our targeting of therapies against that pathology to the right people. We would love to have an accurate scan of alpha-synuclein. Same thing applies to other pathologies. Again, as I said, TDP-43, and vascular disease, inflammatory disease. We need to understand each of these issues at a molecular level specifically to enable targeted therapies and to identify people who can benefit from combination therapies that target not only the plaques and tangles, but the co-pathologies as well. I think that's clearly an approach we need to take.
I think that's such an important point. And, one thought that I was sort of just speculating on in my head in terms of like, if you were to communicate with the translational research community for those researchers who are developing the next generation of, say, proteomics and transcriptomics tools, and what what would you like to see them do to sort of enable these assays, these methods to become more available for clinical trials? Obviously, there's a very high regulatory threshold, and what have you seen as perhaps some of the pain points along that process?
I would take lessons from what's happened with the markers of amyloid and tau pathology. So the biggest breakthrough of the early 20th century was definitely amyloid PET scanning in 2004. And for the first time, we could tell who had Alzheimer's and who didn't. And that in itself is a huge advance in therapeutic research, because prior to that, 25 or 30% of people in our trials didn't have the disease we were treating. That's not helpful for those people. It's not helpful for clinical trial design. We need to see what we're doing. And if we're targeting Alzheimer's disease, we need to see it. We can now see amyloid with amyloid PET since 2004. We can see tangles with tau PET since about 2014. We need to see the other pathologies too. And that will greatly improve our ability to design trials, select people for trials, and target therapy. But PET scanning is expensive and it carries a radiation risk. And the next major breakthrough after amyloid PET and tau PET, and something that in many regards dwarfs the value of PET scanning, are blood tests that show us amyloid and tau pathology, because we can do widespread screening. It comes back to the story of the head. We would never have finished recruiting for this trial without a readily applicable screening test that does not require access to a PET scanner, which limits the population you can screen. It was only through plasma acquisition, and we set up an infrastructure for remote plasma acquisition that allowed us to go out and do widespread accurate screening, pre-screening for the AD trial. That is the way we succeeded in completing enrollment in that trial. By the same token, for omics studies, we need specific and available and cost-effective tools. So omics platforms are obviously data-intensive efforts with multiple dimensions. I think that we need to follow a pathway that is pointed to by omic analyses, but aim for something that is cost effective and easy. So what works best, we can now say, is an immunoassay in plasma. And so we may not know where to look now, and we may need guidance from broad systems-based approaches. But what we really need, not only for clinical trial design and selection of participants, but ultimately for guiding therapy and monitoring the aging population, we need cost-effective blood tests.
So thank you, Paul. And let's take a deeper dive into the multi-omic world, and specifically proteomics and transcriptomics, focus specifically there. Do you think that proteomic or transcriptomic characterization, the molecular characterization profile, might help to reveal disease subtypes or disease progression or even disease staging patterns that are currently invisible to imaging or any biofluid modalities based on ATN signature by them alone?
Yeah, so, ATN was a major development that really helped us think about Alzheimer's disease in terms of amyloid and tau and neurodegeneration. The disease is obviously more complicated, and ATN, I think, is being replaced by ATNX or ATNV, ATNI for vascular inflammatory axes of pathology. And it's going to go beyond that. We're going to need many dimensions of visibility into the disease process. And we need to find markers of those additional changes. We know that the disease is quite heterogeneous, and we can see this in A4 and ahead. In the A4 population, and again, we have the long-term follow-up data, we see that some people progress cognitively very rapidly, even though they entered the trial asymptomatic. We know some things that drive that process, amount of amyloid, level of p-tau217, and degree of cognitive impairment at baseline. So by cognitive impairment, I mean we have sensitive cognitive measures that can pick up differences up within the asymptomatic population. And all of those indicators help us determine the likelihood of rapid progression over the first even couple of years of follow-up. I think that's a starting point. I think that we're far from fully understanding the heterogeneity. And yes, I do think that having measures of the significant aspects of the inflammatory response in brain, the vascular disease in brain, and other proteinopathies are going to allow us to go further than we've gone so far, focusing on amyloid and tau. So I certainly think about that and very much hope to see progress in deepening our understanding of the molecular basis of disease. One thing that I will say is that I think the way you ask that question suggests that we want to identify people for trials who are most likely to progress. And that's a general view. It's one that I, by and large, disagree with. But it's a general view because those are the people who need the most help, right? They're going to decline soonest. But I would just call your attention again to the fact that the earlier we intervene, the more, the greater the impact on the overall disease process. So when I see trial designs that suggest we should select people who are going to decline fast, I always want to adjust the discussion to consider that the people who respond the most are people who have the target pathology, but are in the earliest part of the curve, not the rapid decliners who are at a later part of the curve. So yeah, we should understand what's driving decline. That's how we're going to find candidate therapeutics. I just don't think we should restrict our trials to people who are already on the precipice of rapid decline.
These are some really, really complicated issues, and certainly in terms of thinking about these methods and how to actually start to standardize. I think that that's going to be sort of the subject for some of our future discussions. Paul, we were talking a little bit about where this podcast series could go in terms of really trying to square the debate, and maybe even not so much a debate, but really just sort of a discussion to get other stakeholder viewpoints here. In terms of bringing together all these methods, I mean, we've talked a little bit about, the vast amount of data that's been made available that will hopefully continue to be made available. Internationally, that's sort of a focus for all of us in terms of trying to standardize a lot of this work. Where do you see again some of the challenges and opportunities to really sort of coalesce the entire international Alzheimer's research effort?
Yeah, so first of all, I think we're in agreement here that we need basic science, translational science, and clinical science all to come together to move us forward. And we need data sharing, collaboration internationally. We have to work together. I think it's by working together that we've gotten to this point, and it's through working together that we'll make further progress. That means that each of us should be open to many ideas, not just our own ideas, but we should be listening to each other. We should be exploring every plausible strategy. It doesn't mean that every strategy makes sense, but each of us has to be open-minded and science-guided in moving forward. So I think it's great that we have terrific international conferences that bring people together. I think that the more that we share our ideas and preferably share all of our data and our software and our images and all the materials that we have to bear, and work together, share these ideas and move our understanding of the science forward, that's how we're going to make progress faster. So I think our field does pretty well with all of this. I think that over the years and the decades, we've seen tremendous growth in scientific effort to combat Alzheimer's disease, and that has been wonderful to watch. And starting with your father, Ara, we have made enormous progress by working together and communicating effectively, and all that has to continue. The basic science and the clinical science must continue, and sometimes the environment these days does not seem as conducive as it should be. I think that everybody has to think about the importance of this disease. It is the most feared disease of aging, and we still have a lot to learn, and we have to be open-minded, expansive in our thinking, and then we're going to make steady progress.
I think that's really one of the keys to the entire Alzheimer's research community has been sort of this ability to attract and innovate from many different disciplines and to communicate and really to sort of leverage that knowledge. Ornit, I think we're rounding into corner number 3 on our list of topics.
Yes,
this is
we are there.
Conversation thus far.
So, Paul, I share with you the vision that we have to start a treatment early in disease, very early, even years before clinical symptoms, in order to have a beneficial impact for the patient. So imagine an ADRD clinical trial where plasma or CSF proteomics reveal a subgroup that will be more responsive for a treatment, that despite their similar biomarker for amyloid or tau burden. So what would you need in order to believe that this signal is real and actionable?
Yeah, so I think that there are certainly going to be molecular predictors of response. Hopefully, things that we can see in plasma to make them more practical for widespread use. I think that we know already that, again, that there's heterogeneity in response to effective therapies like lecanemab and donanemab. This is going to be true at the early stage as well, when we won't have clinical features to guide our decision making. We're going to look for biomarkers, for measures that can help us understand who is going to benefit from a specific intervention. And, you know, as I said earlier, I believe that some people are going to be prone to one co-pathology. We define Alzheimer's disease on the basis of plaques and tangles, but, you know, half of them are going to have synucleinopathy. Many of them are going to have TDP-43 disease. Most of them are going to have vascular disease. And I think we're going to see that there are markers of these co-pathologies that influence response to each therapeutic. And ultimately, we're going to select therapies on the basis of a molecular signature. I hope that this is a plasma signature, and I think it will be. I think that we have lots of banked plasma to search through and to look for predictors of decline, but it's really the guidance for effective therapies is going to be a molecular signature, hopefully in blood. So we hope that lecanemab and donanemab are going to have a major impact on disease progression when they're administered at the pre-symptomatic stage, and we'll find out from Trailblazer and from AHEAD whether that's true. But within those data sets, again, we're going to have lots of plasma. We're also going to have all kinds of data. And I think we will be able to determine, to screen large numbers of candidate indicators for predictors of response to that therapy in a trial of that therapy. I think we have to go further and look at predictors of response to therapies that we don't have yet. In other words, which factors clearly are influencing the disease process. And then we'll have trials, and those trials will be conducted in the people who can benefit from those new, not yet identified interventions. So again, it's going to be about keeping an open mind and collaborating, using data we've already collected, furthering our understanding of the molecular basis of disease. That's how we beat it. I want to say one more thing about blood testing in particular, and hoping that our omics colleagues come up with practical immunoassays for markers of aspects of the disease. Ultimately, this can readily become part of the primary care management of the entire aging population. I see people having a blood study related to Alzheimer's pathology as part of their annual checkup. And then when we have signatures and we understand the relationship of signatures to progression along different axes, we will be able to use appropriate therapeutics in those who will benefit from them. So I think that future will be greatly served by having blood-based biomarkers of all of the components of this disease.
And this will move us toward precision medicine, maybe even personalized medicine.
Yes.
For Alzheimer.
You know, that's such a wonderful Wonderful comment. And this is a question, really it's not for you, Paul, but maybe perhaps you've heard things from regulators, from trial sponsors, from other funders in terms of sort of the applications of sort of using things like proteomics and transcriptomics for patient stratification, or possibly even sort of developing very precise clinical pathways for individuals. What have you heard, if anything, from those groups in terms of sort of embracing sort of this more expansive viewpoint? Are they are they a little hesitant? Are they not quite sure what this is all about, or they're thinking about it?
I think regulators are also our partners in therapeutic work in Alzheimer's disease. I felt that from the beginning. I think just as we want to understand the disease, and just as we want to advance safe and effective therapies, so do they. I don't think we think differently about this. And I think that the FDA, other regulatory bodies, they want us to understand this disease as fully as we can. They want us to be able to see what we're doing in regards to every co-pathology of the disease to enable us to safely and effectively treat people to get the best response to therapy. I think increasingly over the years, I've seen regulators be encouraging us to collect as much data as we can to advance the field, even if we're not advancing a specific candidate therapeutic. So I think they see this the way we see this. They want to understand the disease better so we can treat it better. I don't think anything is off the table as far as how we go about this. Again, some approaches are going to be more cost effective than others, but I think they absolutely welcome and in fact urge us to collect very deep data sets that explore every plausible mechanism so that we understand the disease better. When it comes to precision medicine or looking for subgroups that may be appropriate for one therapy or another, there are specific trial design issues there. And subgroup in some sense is a dirty word in clinical trials because that's what leads people down the wrong roads when they use post hoc subgroup analysis to suggest that there is a group that will respond when really they're just identifying that group based on response post hoc and it's not statistically valid. So there are important implications for trial design in terms of specifying a hierarchical approach to analysis that can give us clear answers with regard to subgroups. But that caveat aside, I think we're all on the same page that we want to understand this disease as comprehensively as we can, and that's the way to optimize safety and efficacy in our treatment.
And from your conversation with with groups of the regulators, sponsor, investors in clinical study, from conversation with this group, what are the top barriers for adoption of the multiomics and molecular phenotypes into ADRD clinical trials? What are they pointing out as caveat? What do they want to see in order to move forward with this and implement them into the clinical trials?
Yeah, I think what they want is the scientific validity. So it is a challenge when you're taking a broad approach. You have so much data and all the steps are taken to assure statistical validity of conclusions, but there's a challenge there. A data-driven approach has challenges, just like specific hypothesis-driven approach has challenges, and we need to do everything. So when we see a signal that comes from a broad investigation of proteomics or transcriptomics, we need to be very clear in our validation of that signal using additional data sets, narrowing it down, confirming, and then considering what it means for therapeutic ideas. I think that this can be hugely helpful, but it can also, send us down a wrong path. So we need, again, not to get set in our own ways and convinced of our own theories. We need to be open-minded and collaborate with other groups and make sure we get all perspectives in the way we look at the data. So yeah, we have to, I think in kind of systems-based approaches in particular, we have to be careful. We have to make sure we know what we're doing, and getting multiple voices and multiple collaborators is a way to do that. But everybody thinks that we have to look deeper than, we have to know more than we know now. And that means taking a broad look, and then teasing apart what is real and what is actionable to get us to more effective therapies. So it takes scientific rigor.
Yeah, absolutely. And I think this is the next transition to our
Yeah.
corner 4.
Yeah, I think so. So we're getting towards the end here, and thank you very much. It's always easy to sit here in Washington, D.C., and I know, Paul, we used to be neighbors a long time ago, but it's simple to sort of throw out these very high-minded ideas, such as, what does it look like, or how can we think about perhaps integrating proteomics and transcriptomics into the ecosystem of healthcare, and what are going to be some of those relevant measures in terms of trying to take that, really the multimodal data, and trying to make sense of it. And certainly you've just said that, it's going to take a lot of scientific rigor. It's going to take validation, and it's going to take cross-validation probably as well before anybody trusts or believes what those signatures might mean. And while it's sort of easy to say that this is something to be done, I mean, you've lived the hard road of trying to harmonize data and put these data pipelines together. What can you tell, those people who are getting involved in healthcare policy and planning, particularly for Alzheimer's research? What are some of the big ticket items? Where are some of the gaps right now in terms of trying to fund this infrastructure, do you think? And what would be sort of your recommendations?
Well, this is a hard road, as you're saying. But there's no more important work than the work we do. I think we need to make sure that we are communicating effectively what we're doing so that policymakers and funders understand what we're doing. I think it is going to be most effective if we have diverse and yet unified viewpoints about the importance of deep exploration of the science, collaboration and interpretation of the data, collaboration around the rigor and the validation of findings. And then I think everything falls into place, and the funders, the regulators are on board. Everybody agrees that the cost of this disease is enormous and defeating, and the suffering in the families affected by the disease, which is most families in the world, no effort can be spared. But we need to work together, and if we are effective in working together and communicating and choosing our experiments and discussing broadly our data and drawing inference together, we will be able to communicate effectively, and the results will bring all groups together because everybody agrees with what we need to do. We need to control this disease. Everybody agrees with that. The primary principle is in place, and so we just have to make sure the science is as effective and rigorous and compelling as it can be. And again, that comes through collaboration, open-minded yet rigorous scientific investigation.
Thank you, Paul. So what are your thoughts? Are we moving toward adaptive and modular endpoints, something that change dynamically over time and impacted by different factors? What would that mean for trial design, the conduct, and the analysis?
You know, ultimately, for a drug to be approved, there has to be clear evidence of clinically important benefit. So simply having someone enrolled in a trial with an elevated p-tau 217 and having a therapy that changes that p-tau 217, that's not enough. That's not enough. But if our understanding of the disease is richer than it is now, and we see that p-tau 217 collaborates tightly and predicts accurately the clinical ramifications that are still down the road, then it becomes an element of trial design that can lead to approval. And the FDA has a mechanism for approval based on findings that are not in themselves clinically meaningful if they are reasonably likely to predict a clinically meaningful benefit. So you can apply that principle to a molecular signature and what you can tell from that, or a specific molecular measure. If there is robust data that leads to a view that changes in this mechanism and this measure are reasonably likely to predict clinical benefit, that's a path to accelerated approval with continued data collection to nail down the long-term benefit after the drug is in clinical use. So that's a pathway. It's not an easy pathway because correctly, regulators have to be convinced by robust and clear data, but there is a pathway.
Thank you. And this takes me directly to our final reflections. As a leading participant in the Leon Tau Symposium series, you may recognize this next question. If you had to name the top 2 or 3 enablers for making proteomics and transcriptomic informed trials real, what would they be? In the infrastructure, funding models, policy levers? Just give us your insight.
You know, my thinking is that we shouldn't think in terms of just policy or just funding. We should think about the clinical science coming from the basic science. And when the science is right, then everything falls into place. I think, should we have a molecular marker coming out of an omics platform that is clearly predictive and targetable, then I think everybody gets on board. So again, it's about doing the best science, having the best environment for productive science. When we have it, everyone will come together. So I'm not so worried. Yes, we need more infrastructure. We need more laboratories. We need techniques that are more cost effective and more widely available. We could list many, many things we need, but the science will drive our progress and everything will come together when we get the science right.
Thank you for highlighting the importance of the science. It's very encouraging.
Yeah, it really is, and I think it subsumes so much of what needs to happen, and I think that's really sort of one of the outcomes for this series is to start bringing together the different voices, the different viewpoints, and actually it's more different perspectives to sort of coalesce around ultimately what good looks like. At least the last question I have for today before we sort of wrap up is so much of the questions that we've asked have been around the science, and we've sort of identified some of the challenges in doing that. Yet it's really difficult to sort of divorce that from the fact that ultimately for a lot of the drug studies and therapeutic studies that are conducted, we rely on industry and industry partners. And those industry partners have their stakeholders as well, which primarily are the capital markets. And increasingly, we've all seen at the scientific meetings an increase. I don't know if it's logarithmic or linear, but definitely a lot more investors and investor bankers come to hear the readouts from various drug studies to get a sense. But how can we actually maybe think about getting them a little bit more ahead of the curve in terms of thinking about, these methods and perhaps these ways to sort of adopt future clinical trials and to sort of maybe perhaps de-risk or at least open up the ideas that there are these different methods and we really should consider putting them into trials. Um, what have you seen that has worked, and what else maybe could we think about trying to do?
Yeah, so I think the model for moving forward is the model that has worked for us, which is academic industry partnership. I think the public-private partnerships have been hugely successful. I'd point to ADNI. ADNI has tremendously advanced our therapeutic search and is to an important extent responsible for our success. And ADNI came about through academic investigators collaborating with industry investigators and funders, all working together. Trials like AHEAD is a public-private partnership with industry and academic co-funding and funding from philanthropic groups and everybody coming together. I think that the risks and the new ideas are often best explored in the academic sphere. But as they advance, we need industry partnership and we have to work together. And I think that's valuable to industry. And that's why you see more industry academic partnerships than we had in the past. I think taking the academic environment as one that can explore high-risk ideas and is not directly reporting to stockholders, say, we can take a deeper exploration of new ideas. And the pre-symptomatic trial design is a good example. That's something that I don't think any company could do on its own, but multiple companies embraced as an academic industry partnership. So I think the way forward with new ideas and with omics investigations is to work on this broad collaboration that encompasses basic science and clinical science, academia and industry, everybody together, and we make the best progress.
Interdisciplinary partnership is definitely the moving forces to make things happen. Then when we have everything ready, we're looking into health system and the actual trial site that will be doing it. What you foresee, how can we get them ready for what is coming from basic and translational science advancement into the actual site of the clinical trials and basically accessibility to the patient and how the patient will accept it?
So I can tell you that we are hugely helped by the development of blood-based biomarkers. Blood-based biomarkers give you, are just readily applicable everywhere internationally. So unlike PET scans, for example, a blood test can help you with trial design, and it can be used at any clinical site and enables us to reach more representative populations and to have more aspects of our trial design be decentralized. And this is a great path forward. Lilly has really broken ground in this direction in their Trailblazer-ALZ 3 trial, which heavily relied on blood testing and did only limited PET scanning. And I think we'll learn a lot as we share as Lilly shares the results of that study. But I think that including methods that are readily available everywhere, like blood-based methods. Or Lilly also used central raters for cognitive and clinical assessments so that trials did not need the expertise at that level. I think this applies, can apply to many new areas of trial science where we can take advantage of what we learn in the laboratory by using specimens that are readily can be readily obtained anywhere.
And maybe some educational session or outreach to the communities. We work together also in collaboration with the patient can be helpful in order to implement all of this at the actual clinical trials.
Yeah, and you know, you touch on an important issue that we really haven't discussed, but yeah, the partnership is not just academia and industry and funders, it's the families and the patients. Our field has moved strongly in the direction of including the viewpoint of participants and families. We have a participant advisory board that helps us design our trials and keeps the interests of the participants in mind as we set up every procedure and every study, and that's important. That's important as well.
Multi-level partnerships.
Right.
Okay.
Well, Paul, thank you so much for joining us today. You've been very gracious with your time, and this was, I think, a really great way to kick off the series. Thank you, Ornit, as well, and thank you to Frontiers.
My pleasure.
And next, thank you. It was a pleasure. Great conversation. And next time, we are going to dive into some of the analytical and infrastructure barriers with Dr. Michael Watts, looking under the hood at the multi-omics data pipelines. Thank you all, and have a great night.